for those of you who are new, just a simple figure. Mycetoma is caused by bacteria or fungi which are introduced into the sole of the foot by a small trauma. There they form granules called grains, and the lesion starts to grow further inwards till you get these massive lesions, which you can see over here. In real life, it looks like this. And then inside the tissue, even the bone is destructed by the fungus. Typically, eumycetoma at the moment is treated with a combination of itraconazole and surgery, which means that itraconazole is given for 6 months, then the lesion is surgically removed, and at least another 6 months of itraconazole is given. It cannot be treated with itraconazole alone, as simply the lesion will not disappear and it remains quite stable in this first 6 months. So there's a need to identify a novel treatment for eumycetoma, and preferably one where you don't need the surgery anymore. So as Matt mentioned before, there are 2 different routes of identifying novel drugs for mycetoma. The first one was by screening drug libraries, and by now we screened more than 8,000 to 9,000 compounds for activity against Madurella mycetomatis. But you could also identify novel drugs by gaining insight in the disease development. And that's the part I would like to discuss for today. So if we want to study if a drug can penetrate a mycetoma grain, we are stuck with in vivo models because grains cannot be formed in vitro. And luckily, we developed a few models for that. And the invertebrate model in Galeria melanella actually produces grains which resemble human grains. So how do we do that? So we infect Galeria melanella, the invertebrate, monitor survival, and as you can see, within 8 days, all larvae will die. And at several points, you can actually look what is happening inside the larvae. So we did that, and we saw that already 4 hours after infections, the early signs of a grain can be noted. So here you see these melanized structures, which is the Madurella mycetomatis grain. And in the case of Falciforme senegalensis, it takes a little bit longer. Then in the case of Madurella mycetomatis, cement is forming, it will be encapsulated, and finally an immune efflux will take place. For Falciforme spora, the similar process takes place, but it does take a little bit longer. And both are the most common causative agents for human mycetoma. So if you wonder if this invertebrate can really form a grain which looks like the human grain, well, actually you see similar features between the species. So Madurella mycetomatis has the cement material throughout the grain. We see that in the Galeria melanella model too. With Falciforme spora, you see a more melanized rim and a little bit more loose center. And in Galeria melanella, that's the same feature we see. So the Galeria model, although a very simple model, can produce grains which resemble the human grains.
I have a
So,
question.
yes.
Does that mean that Madurella mycetomatis might in the environment be an insect pathogen similar to something like cordyceps or whatnot?
No, that does not have to mean that. Galeria can be used for many infections, also infections which are not prone to insects. It's simply the Galeria has an innate immune system which resembles our own innate immune system. So probably the grain is the result of the interplay between the two.
Ah, interesting. So not much is known about the ecology of these organisms then.
No, actually, Madurella has never been cultured from the surroundings. So we don't know the natural niche of Madurella.
Very interesting.
Okay. So what I was saying, in this model, we can actually just follow the grain development over time, and we use this to isolate proteins and RNA from different time points. So a very early grain, an established grain, a mature grain, and a very mature grain. And we can then, using proteomics or transcriptomics, actually map the processes leading to grain formation in both the host and the pathogen. Furthermore, we saw that based on the proteins and transcripts observed, it appears that Madurella mycetomatis remains metabolic active throughout the 7-day observation period. So that's also important for drug discovery. So based on the analysis we did, we made this model, and we see here the roles of the host in each phase of the grain formation and the ones of the pathogen. And for drug discovery, we will focus mainly on the pathogen. So what do we see? In the first stage, we have an increase of adhesion and transport proteins. So we took a closer look at which proteins there are, and we found the transcriptionally controlled tumor protein, fructose bisphosphate aldolase, and pyruvate kinase. And in this study, we mainly looked at the antigenic properties of these molecules. And we do see that patients have relatively high antibody titers, and Dutch control have low titers. However, these drugs are housekeeping, proteins are housekeeping proteins, and they bear quite a lot of similarities with their human counterparts. It's actually known that for the fungal TCTP can be used to stimulate the human immune system in a similar fashion. So it's less likely to get a specific compound interfering with these proteins. We also found that there was zinc and iron acquisition important. So with zinc, we found that the zincophore system was expressed, and with iron, we saw siderophores being produced. And this is together with EMAT, this work. So if we look at Madurella mycetomatis, there's a pathway of siderophore biosynthesis, which is quite similar to the one from Aspergillus fumigatus. And we found siderophores transcripts expressed present already 24 hours after infection, so in the premature grain, and they remained there till the late grain. Furthermore, what we saw when we gave the larvae an iron overload, the grain was not compact anymore. Hyphae started to grow outwards, so the metabolism inside the grain was actually changed. So the siderophores might be a potential drug target. What is also known is that that a company actually made a siderophore molecule which became antifungal. So they actually used it as a Trojan horse to make it an antifungal agent. What we also found was that, especially in this phase, the stress response was upregulated. So there are several factors in the stress response, but we focused mainly on the kinases. So what one of the other PhD students did, so what Mickey did, is looking in the genome of Madurella mycetomatis, and he found 132 predicted kinases. 68 had no human homologs, and 21 were shown to be essential in other fungal species. Of these, 4 had no human ortholog and were found to be essential. So then we looked with EMAT at the expression during infection, and 2 were actually expressed. So we looked into which genes these were, and they appear to be both as part of the cell wall integrity pathway, which you can see over here. So this might also be a very important pathway to target because it's essential, but also it plays a role in azole stress. So this might be a good candidate for combination therapy. Then we found the last part that the grains are melanized, and fungi use DHN melanization. So, and in the case of Madurella mycetomatis, again, we saw expression of the melanin genes during infection. And the melanin pathway mainly in use by Madurella mycetomatis is the DHN melanin pathway, for which actually known inhibitors are known. So if we then look at the normal grain, which is depicted over here, and we expose it to carpropamide, this molecule inhibiting this part of the pathway, we got a non-melanized Madurella mycetomatis grain. So we wondered if this would actually help. And what you do see is when you treat it with carpropamide, you get enhanced survival. So it looks like this non-melanized grain is less pathogenic than the melanized grain. And since the melanin is no longer present, we also combined it with itraconazole and we saw a prolonged larval survival again. So also the melanin might be a good drug target. So summarizing the pathways we came across in both the proteomic and the transcriptomic experiments, we saw that there was some inhibition of adhesion to the host, which was important, vesicle transport, cross-linking the extracellular matrix, preventing zinc and iron acquisition, etc. And we then determined if they were fungal specific, yes or no. Well, most of these are not fungal specific, but the trehalose pathway is not present in human. So yes, it's relatively fungal specific. The warrenin body is a specific fungal organelle not found in any other species. And melanization, the DHN melanin pathway, is also fungal specific. For most of the non-fungal specific pathways, there are inhibitors known. For the zinc and iron acquisition, yes, there are some fungal specific molecules known, such as GR2397, which was shown before. Interfering with kinases, yes, there are fungal specific kinases. There are also, of course, general kinases shared between human and the fungus. But in our screenings with a large drug library, we found at least one which was able to inhibit the kinase of Madurella mycetomatis and also prolonged larval survival. For melanization, there are also some fungal specific inhibitors known, and we also know that these will enhance larval survival. And that was a very short introduction of the years work we've done for the past decade.
That's great.
So are there
Thank you.
questions?
Hi, Wendy. I do. I mean, maybe for you and Matt. Is a host also protein targets also being looked at, or not really? You're not looking for host-directed therapies in this SGC kind of strategy?
So we know the responses in the host, so we mapped that here as well. As you can see, that's the blue part, but I didn't look into compounds inhibiting the host for now. It's something we could certainly do in the Galeria melanella larvae too, but there we are stuck with that the larvae does have an innate immune system similar to us, but not an adaptive immune system. So how comparable it is, I don't know. But for some molecules, we could use this model still to figure things out.
Okay.
Definitely relevant and interesting. Yeah.
And this will be kind of okay also for the SGC. We could even use that. Okay. Yeah, it's just because the NCI is looking at more like HDTs for some of the other neglected diseases. So I guess this will also be something interesting. Yeah.
Yeah.
Has chitin...
And especially also for the mouse model later on.
Sorry. Has chitin synthesis been looked at? I know that it wouldn't work in the caterpillar model, but mammals don't synthesize chitin. So has that been looked into?
Yes, Dunya screened recently a library where all fungicides either used in agriculture or in human have been tested. We do have chitin inhibitors there too. In vitro, we didn't see that much, but we also didn't see that much with the Ibraxa fungerp, for instance, it has a very high, relatively high MIC, but it did prolong larval survival. The chitin inhibitors we didn't try in the larvae because we will need to establish the effect on the larvae itself first, because the larvae also forms chitin. That's also the reason why trehalose wasn't tested before. Trehalose is not found in human, but it is found in insects too. So that's also a little bit more difficult to study in the insect.
Thank you.
Wendy, I have a question. Really excellent stuff there. I noticed that you highlighted the warning body as a potential target right on your very last slide. Do you have evidence that the function of the warning body is critical for infection?
No, but we noted both in the proteomics and the transcriptomics that it was highly upregulated during grain formation. So since the warning body, for those who are not familiar with it, seals the septal pore when a fungal cell is damaged, it prevents leakage of the cellular component when this damage is done. So if you would find an antifungal agent which damages the cell wall and cell membrane, and you combine it with an inhibitor of the warning body, that would lead more rapidly to killing, I guess. At least that's my hypothesis. But it was one of the most abundant proteins found actually in the grain.
Right. I mean, just to highlight that we recently published on a protein kinase called YAC-A, and we found that regulates septal plugging by the warning body. And we've shown that we can target that kinase with compounds, beta carbolines.
That would certainly be interesting to test.
Sure. Yeah, absolutely. Yeah, we can provide that to you, those compounds to you if you want, or at least provide.
Yes, if you are willing to do that, yes, please.
Yeah, sure. Yeah, no problem.
Am I right in thinking that that is a complex of proteins rather than one thing? Apologies for my ignorance.
Yeah, there are quite a few proteins that are associated with the warning body. There's a tether that tethers the actual plug. There's the plug itself. There's a lot of proteins that are associated with regulating how that protein functions and how the plugging occurs. So yeah, it's not just a single protein.
Right.
But at least the kind is certainly a target that's amenable for screening.
Yeah, that's very interesting. Okay. All right. Fantastic. Thanks, Wendy. So, Michael, did you want to say something about what you've been doing in a related fungal pathogen?
Sure. So I'll keep it very brief. But our organism of interest is Aspergillus fumigatus, which many of you will know is a critical human pathogen. We were funded by the Wellcome Trust a number of years ago to generate a genome-wide knockout collection in Aspergillus fumigatus. And what that meant was that we went gene by gene and disrupted or removed each gene from the genome. Through that process, what we've been able to gather is a set of information of genes where we cannot get a viable non-mutant. So theoretically, these genes should be essential for viability in Aspergillus fumigatus, given the screening process that we went through and the selection. So obviously we select on hygromycin, so anything that's hypersensitive to hygromycin will come out in that screen too. So what we've got is an enriched set of around about 3,000 genes that are likely to be essential for viability. So that set is something that you could mine within your project to prioritize drug targets in mycetoma. So it probably would be relatively straightforward for this group to identify targets in your organism based on homology to the hits that we've got from fumigators, and use that as a start point for prioritizing your targets. I note from some of the reading that I've done, Wendy, that you've developed a process for generating gene knockouts in mycetoma as well. So I guess for any target, what you would want to do is validate those targets by gene knockout or promoter replacement or something along those lines.
Yes, that's actually also a part of the postdoc project for Tim. He's the one going to make the knockouts.
Great. Okay. Well, we're more than happy to share that information with you if you want to think about prioritizing your targets and whether whether you would consider them to be druggable at this stage. So just because they're essential doesn't necessarily mean that they're going to be a druggable target. So there's quite a bit that we can do in advance of even trying to generate non-mutants to prioritize those targets. I think in a drug discovery setting, you're always looking at projects that can generate sufficient income to warrant the development of a novel antifungal. The difficulty with mycetoma, of course, is that it is a relatively niche pathogen, and that can be difficult. One way to potentially address that is to target enzymes or factors that could be critical across a spectrum of fungi. So going for something that's a little bit more broad spectrum, but making sure that you include mycetoma in that spectrum, and therefore you're developing compounds that may have activity against some of the the big boys of the fungal world. So
yeah, absolutely. It's I think it has to be that kind of plan. Yeah. Otherwise it's very difficult to attract the kind of funding. For the 3,000 or so likely essential proteins, do you have a sense of the map of how many of those have close human orthologs?
no is the answer to that, because we haven't mined that data specifically. So I suspect there will be quite a few with close human orthologs, but again, it's what you consider to be close, and how you judge that is not always straightforward. We had these discussions many times about what does close mean, and if something has 50% identity over 40% of the protein or the active site, what are you looking at? So those calls are quite difficult to make in terms of filtering. Yeah,
absolutely. Yeah, no, we have I may have mentioned we have a malaria project where we're looking at a human WDR protein which has a small molecule binder. It's a micromolar binder, but it's a binder. And there is a kind of orthologous malarial plasmodium protein, and if you look at the homology, it's about, I don't know, 60% or something. But in the site where the small molecule is binding on the human protein, that versus the plasmodium protein, there's a single amino acid difference, right? So it's perfect as a starting point, because you think, well, we're likely to be able to tweak the molecule to get selectivity. But if you just look at the absolute figure, then yeah, it's not high homology at all.
Yeah, indeed.
And sorry, it's just again a sort of approximate figure of the 3,000 or so. I mean, is it the case that there's a lot of unexplored territory there? So classes of proteins that maybe have been not well explored, proteins whose function are unknown?
Yeah, absolutely. Yeah, there are. I couldn't give you a number right now, but there are definitely quite a few proteins of unknown function in that collection.
Okay, that's great. All right, well, we need to have a look at that, and I guess yeah, Wendy, I guess it's a good one to think about for your project, Tim, about something that we might be able to want to validate against mycetomatis. All right, thank you, Michael. Louisa, would you want to say something about DNDi's approach maybe in this area and priorities as far as you're concerned for new targets?
Yeah. So again, I'm sorry for my colleagues wouldn't they were not able to join today. There was at the same time the reviewing meeting in Geneva. But anyway, so yeah, so I was just looking now. So I'm mostly working on Chagas disease and leishmaniasis, but I was checking what we do for mycetoma. And we unfortunately don't do a lot for drug discovery. What we do is basically under the umbrella of the mycetos. But we are going to maybe slightly moving away from our focus from Chagas disease and leishmaniasis. Now we're going to schistosomiasis, and we do have a whole team for mycetoma. And so I guess the NDI role here will be more like in the compound, when you get the proteins maybe, and then we can maybe access libraries or facilitate or help with logistics and everything. But in terms of targets, what Jean-Robert, I know you know Jean-Robert, Matt, he said that it would be difficult to do it. And I guess he didn't take into account the whole like understudy or never study protein targets. But what he mentioned was just to use whatever is known in other fungi, and then see if we can see if there is a closer ortholog or a closer protein target in mycetoma, but I guess that's not what you're looking at. But yeah, so I guess we don't do a lot, but we are probably okay to support whatever activities are ongoing on that area.
Okay, great. And you mentioned something about human factors. Did you want to say anything more about that? Because that's something that's really interesting for a couple of reasons. One is that they're not, they're not fungal targets, but also that the assumption is that they're more difficult to develop resistance against, right? Because they're human.
Yeah. So I guess again, I'm not the expert here. My colleague who was supposed to join the meeting today, Stefan, is, and I know he'll be very interested. Yeah, and then he'll be very interesting to know about this. That's why I was asking maybe to pass on to him. And Wendy, I'll be happy to have your presentation to share, if that's okay, to share with my colleagues, because it's something that the NDI is looking at for everything now. So we are doing a lot of HTT research, and I'm sure that we'll be able to support that too, if that's something that it's interesting for mycetoma, for the
sure, absolutely, yeah. I was going to present a couple of slides.
Lisa, the presentation is also recorded, so they can also listen.
Yes, I'll send.
That's easy.
Yes, absolutely.
I was just going to show a couple of slides about the screening methods, just to make sure everyone was on the same page. But Imad, I guess I just wanted to make sure that you had a chance to come in about anything that you'd heard today about targets that might be interesting. And no pressure. If you'd like to say something, that's fine.
Okay, thanks, but I'm fine. You can go ahead, and I'm not prepared. Cool. Yes, yeah.
No, that's fine. No, there's no need. Yeah. So the two the couple of things I was going to say. So this is kind of standard SGC stuff about the value here of trying to stimulate activity against targets that are not well known. And it goes back to the analysis of the kinase field and about, which kinases were being looked at. If you look at the full panel of human kinases, lots of activity in a small number of targets at this very dramatic hockey stick shape. It's easier to get funding for targets that are known to be hot, and it's easier to publish big papers on things that are known to be important. And so it often leaves lots of targets not well explored. And this has been the case for the last 20 years, basically throughout, except, let me just do the pointer thing, except with some exceptions here where, between 2000 and 2017, there have been little bumps where some kinases have suddenly become more interesting to the community. And it doesn't really matter where you look. This is worldwide, this is Canada, but it doesn't really matter. It's the same sort of shape where lots of targets are unstudied or understudied. And in one field, as a zoom in to the nuclear hormone receptors, there are some here, and again, the number of citations per target is very, very skewed, with some targets being essentially ignored. But it's kind of interesting that over time, some do develop these little bumps, and suddenly these molecules, these proteins, these targets become more interesting for researchers. And you think, well, what is it that has led to this happening? Why these ones? Is it because they're suddenly found to be incredibly important or not? And you do the correlation with proteins where potent small molecule selective inhibitors or chemical probes have become available to the community. And these are the ones that have had those things become available. And suddenly with that molecule, which is able to shut down a protein in a cell and allows you to study it effectively and selectively, you find that the activity goes up, right? Because suddenly you can figure out what it's doing and you've got a control situation. So the idea of developing public domain small molecule inhibitors or binders or disruptors of proteins leads to an increase in the understanding of what that target does. And that essentially is the SGC's mission for the last 20 years and essentially why all of this is public domain, because the idea is to share these resources with everybody to allow people to have a research tool to understand biology. Downstream, of course, then you can develop those public domain compounds into new chemical space, including in the private sector, to develop drugs. But initially, the tool compound is public domain without any restriction on the IP and without any restriction on the use. And so, you can do the same analysis here where you've got chemical probes available on the left there and not on the right. And it really does. It's a strong correlation between those things. So the idea here is that, you do the screening approach, you try and develop a small molecule inhibitor or binder so that people can understand what the target's doing. Therefore, understudied or not studied proteins are by far the most important, or by far the most interesting to the SGC. And just quickly, the two screening methods which the SGC has been publishing on and has preprints. One is a very interesting thing called DNA-encoded library combined with machine learning. So DNA-encoded library is where you have a molecule attached to, your molecule is attached to DNA and you do the screen and the binding screen and you isolate the binders and the not. And then by sequencing the DNA, you figure out which molecules are binding and which are not. And because these are just molecules that are labeled, you can screen billions of molecules at the same time in a real mixture, so it's very efficient. The challenge is to build a library and to do the decoding of what the molecules are, and to deal with lots of false positives that arise because every small molecule has a whacking great DNA chain attached to it. So this paper from what was a Google team did a very interesting thing where they did a DNA encoded library screen and isolated the binders and the not, and then used that data to develop a machine learning model, which was predictive of which molecules should bind to this given protein, which you could then apply to a commercially available data set like enamine of 40 billion molecules and predict molecules that should bind a protein. And this remarkably worked, despite the fact that the training set and the library of molecules that's available are not necessarily the same. So it's quite remarkable. And so this has been done on multiple targets now. So multiple novel protein targets where it's the same idea that you get you do the experiment, you get the data, you develop a machine learning model, and then you use that to nominate some commercially available molecules, which you then buy and screen as binders of the protein for experimental confirmation. So it's it's a very interesting area, and even more interesting, you don't need the original data set that comes from the DNA encoded library. You don't need the structures of the molecules that bind. All you need is a sort of abstract fingerprint of what the molecules are. So you can have a proprietary library being screened and public domain data being released without that public domain data revealing what the library was, which is important from the point of view of commercial sensitivities. And so this ChemArchive preprint at the bottom here is what's been going on in this area. And sure enough, small molecules that can be purchased are being found. In this case, from a set of only 50 molecules that were ordered at about $100 a piece, found some molecules that are reliable, micromolar binders. So it's not a drug, right? But it's something as a starting point, which can then be confirmed with crystallography. So you get a binder of the protein. So it's quite interesting in the sense that you have to spend a lot of effort doing this screen in the first place, but those are available commercially. You have to do a lot of sequencing, a lot of data crunching. You then have to develop a machine learning model, which of course a lot of people are very delighted. I couldn't do it, but a lot of people really want to do it. And then you only have to order a small number of molecules at the moment in order to have a good chance of getting a binder which is confirmed in a biophysics experiment. So, ideally doing this with a mycetoma protein would be sensational if we could do something like this. And then the second method of two, this is the last couple of slides, is intellectually much simpler. So you have a protein which is immobilized, and then you mix that with a library of about 1,000 diverse molecules, again, all commercially available, and things bind, and then you separate that out from the non-binders, and then you denature the protein, and you see which molecules by a complicated LCMS experiment. Essentially, you see which molecules are enriched in the set where the protein was. So you can determine a marginal enrichment of molecules that have been bound to the protein. And again, this has been running very well in a lab that's running the SGC in Toronto, where about 8 proteins at the same time are screened versus this library of about 15,000 molecules or so. And in most cases, a handful of molecules are found to be enriched. And so you then take those and check that they bind by something like SPR or something else, and then do X-ray. And a bunch of molecules have, again, been shown to bind to these proteins, which they're not well understood. They've been used as model cases for the SGC, but some of these have been in the past resistant to productive screening campaigns, but are yielding, again, sometimes double-digit micromolar binders, but these are ones which have been confirmed by an orthogonal assay, and again, by crystallography in some cases. And there's a really nice preprint at the bottom there about the work that's been done on this stuff, including a very cool thing where if you have racemates in the library, the LCMS method, which is applied to essentially the supernatant from the denatured protein, can in some cases tell you if there's been an enantiomer enrichment in one of the binders, and that kind of gives you an internal check that it's probably some kind of real binding event, because otherwise why on earth would you get enantiomer enrichment? So this has been going pretty well in the sense of screening about 7 or 8 proteins a week so far, and that's just as a sort of pilot study. But this can be deployed again if we had a pure sample recombinant fungal protein with a his tag. It can be deployed against that, and if the hits are confirmed, it's fairly straightforward to buy a small number of analogs of these compounds to get a preliminary sense of SAR. Like I said, without having to go in the lab and make them. Eventually, of course, you're gonna have to start making some molecules, but initially you can recheck the supposed active. You can do a little bit of SAR by catalog to give you a sense that you're onto something real. So those are the things I'm talking about, and as I said, the idea is that these would be deployed against targets that are novel. That's sort of the quid pro quo. So yeah, if we manage to isolate some samples of something recombinantly, we can certainly deploy it against that. And even though the SGC is a charity which does really prioritize human proteins, the potential impact for things like infectious disease, understanding microbial systems and things like that is obvious, and it's definitely going to be part of the next few years of SGC activity. Anyone have any questions about that? Hopefully it's fairly simple, and it's all contingent on actually isolating some recombinant protein at some point.
Yeah, Matt, as demonstrated in the plot earlier, we made recombinant fructose bisulfate aldolase, pyruvate kinase, and transcriptionally controlled tumor protein. But we made those recombinantly in E. coli. And of course, a fungus also has some secondary structures, sometimes with which are not expressed very well in E. coli. Sometimes you need a yeast or you need another fungus to express those. I'm not sure if we can tell beforehand if the protein will be correctly fooled. And if it's not correctly folded, you will get differences probably from the natural protein. So how do we go about that?
Good question. I'm very much out of my depth on that, but I know a lot of clever protein scientists who will have an answer to that question. So yeah, I can pass that on. Yeah, doing this kind of prediction of how to do the protein production is a key question for lots of these novel cases. Yeah, so
because sometimes we got in issues when the tag was cut off, and when the tag remains on, it was folded okayish.
Yeah.
So yeah, it will depend on a lot of things, I guess, if the protein will be correctly folded. And some proteins are very easily to be expressed, also in E. coli, and some other proteins might be more difficult to be expressed because they might be toxic to E. coli, or simply don't fold correctly, or are not expressed at all because they use different codon. There's a difference in coding usage.
Yeah, so it's E. coli and mammalian and insect are the three systems that are traditionally used. Increasing the amount of mammalian cell expression, I think, is going on. But yeah, it'll depend on what you're trying to make. Yeah.
Yeah, and fungi, they also use Pichia, a yeast, because then you have at least the sugar structures relatively similar.
Right.
Matt, do we have a timeline? Like, what is the perfect, what do you kind of plan for this in terms of dates and timelines? And I know it all depends on actually finding the protein and expressing the protein and everything. But
yeah, sure. I mean, I think it's just one that we need, So imagine that you isolate one and you conduct the screen and you find a binder, and then you confirm that it's a real binder. With some decent value. So you've got something like that. And then you nominate another set of targets that you think are diverse and interesting and understudied, but you validated the concept.
Okay.
Then I think you only need to do one for a realistic grant.
So the goal is kind of to validate this whole kind of platform to see if the screening okay. Gotcha.
Yeah, because I mean, with the resources available, we can do a pilot, but we can't fund the whole thing, right? So the idea is to demonstrate the proof of concept and then go for funding. But as Michael said, if you can show that the targets are of more relevance to other pathogens, also, it just massively increases the chance that we might attract funding for it.
Yeah, great.
Yeah, and maybe it's good to make a top 5 of proteins which will be of interest, because in case you cannot purify it from the fungus itself and cannot make it recombinantly, at least you have a second choice to look at.
Yeah, that's a good idea.
The IP situation is such that the work, it really is about trying to show that these proteins you can find a binder of the proteins, and that you can then most likely find a small molecule that is potent enough that develops a chemical probe and allows you to study what the molecule does. All of that is public domain. And the point about that is that if anybody then, including us, of course, wants to optimize the molecule actually as a drug, they're free to do that, right? There's no restriction on that. That's kind of the point. So we're free to do that, and companies are free to do that, whoever feels they have a shot at doing that. So the work could be of potential value for industry as well as our own effort here.
Gotcha. And just another logistics kind of thing is, do you have already kind of funding source that you would like to apply, maybe if you do get that, or that's too soon?
Well, GHIT is the obvious one.
Yeah, that was what I was thinking.
But those are usually very large. So that's what I was thinking. Unless you have multiple targets and a real sense that it's going to work, we wouldn't be in with a shot really. But
GHIT usually they require a Japanese. So are you thinking about that too? Japanese partner?
Yes, we're thinking about very much. But we would need to cement that relationship much better. You need us, you need a Japanese partner, and you need an NGO, right?
Yeah.
I was hoping DNDi could be the NGO.
Yeah, no, of course, yeah.
Don't want to make assumptions.
I was just thinking about that. Of course, GHIT already has a history of that, and ASI and some others. So I guess it wouldn't be, it wouldn't be too hard of a conversation to start, I guess.
Yeah. Michael, sorry, I didn't see you had your hand up.
Yeah, no worries. I'm just wondering, in terms of prioritizing your targets, if you consider going for the set that we have of 3,000, it's quite a large number. So, how would you think about prioritizing that? Would you, for your proof of principle, would it be sensible to look at an enzyme rather than a structural protein? So the enzymes are going to be probably easier to make, probably going to be more soluble, and probably going to be able to make an assay. I say probably in all of those things because there's no definite, right? So if you are going to prioritize, I think it's useful for the group to think about exactly how it's going to prioritize the targets. Why go for one rather than another? And I suspect that ease of protein production may be one thing, as well as the requirement for being essential for pathogenicity, or maybe even better, essential for growth in mycetoma, because they would be easier to screen. Once you have your drug, you don't need to do a pathogenicity screen. Then you can just look at the activity of the drug against the bug.
Yeah. Yeah, I think that's right. And I think a suite of things will be needed, but an enzyme makes a huge amount of sense. There's an argument to be said for going after something which has a little bit of precedence as a family, because you may have some initial chemical matter that you could go after, but I don't know. Yeah, it's that essentially you've asked the key question, right? What's the nature of that panel of potential targets?
Yeah, and the requirement for this group probably now would be to work out how they're going to prioritize. So that might be a key outcome of this meeting. How do you want to prioritize those targets? I'm really sorry, I have to go now.
No worries. Thanks for coming very much. Yeah, really great. Thanks for your time.
But please get in touch with me if you want to get any more information, or if you want access to the essential gene set from AsperGillus.
Michael, can I just ask you one very quick question?
Sure, Wendy. Yeah.
If we do find a protein which might be of interest for mycetoma, it might be good also if you could look at your knockout or express the protein also for Aspergillus, so that we could target both species simultaneously. Yeah. Also with the idea of making it an antifungal agent, not only for mycetoma, but for other fungal diseases too.
Sure, absolutely. Yeah, no problem at all.
Okay.
I'm happy to.
Thanks, Michael.
Okay, everybody. Great to meet you all, and see you later.
Too. Thank you.
Bye-bye.
Thanks. All right, I also have to run. Does anybody have any nano additions to the meeting they want to share quickly? Or we're going to have to think about follow-up about how we're going to do exactly that prioritization.
No, it was very interesting. I'm going to leave as well, sir. Thank you.
All right. Thanks for coming. All right. Thank you, everybody, for coming. I'm sure lots to follow up. We're asking the key question, I think, so we'll defer that conversation to a future meeting. All right. Thanks all.
Thank you. Bye-bye.
Bye.
Bye. Bye.